Welcome everyone to this second virtual Oskar Fischer Lecture, which is the fourth in the series that began in early 2020. I would also like to thank all the attendees, not only those at the schools and colleges throughout UT Austin, but also from other universities and organizations elsewhere across the nation. A bit of history here. This series came about through the vision of Dr. Jim Truchard, who wished to keep the accomplishments of Dr. Oskar Fischer alive through the endowing of the special lecture series. Our previous lecturers have been experts in the field of neurodegeneration. The first lecturer was Bruce Miller, who's from UCSF in San Francisco, and he is an expert in frontal temporal dementias. Our second speaker was Tony Viscore from Stanford University. Dr. Viscore investigates age-related cognitive decline and has discovered a serum factor, circulating factors in the blood that modify brain structure and function and could inform treatments for dementias. And the third lecture was from David Holtzman, who's the chair of neurology at Washington University and leader of an Alzheimer's center there. He's done fundamental research on how sleep influences metabolism of proteins such as A-beta that are thought to be part of the pathology of Alzheimer's disease, and has studied the importance of sleep on cognition and general health. Today, I would like to introduce you to our fourth speaker, Dr. Beth Stevens. She comes to us from Harvard Medical School, where she is an associate professor in the F.M. Kirby Neurobiology Center at Boston Children's Hospital. She's also a member of the Broad Institute and the Stanley Center for Psychiatric Research, and is a Howard Hughes medical investigator and a member of the National Academy of Medicine. Dr. Stevens has won many prestigious awards, but one that I would highlight particularly is that in 2015, she was chosen as a MacArthur Fellow and got one of the MacArthur Genius Awards. She also has shared the National Alliance on Mental Illness Research Award with Stephen McCarroll and Michael Carroll in their 2016 collaborative work on neuroimmune signaling and schizophrenia. Her research focuses on understanding neural immune interactions and how they sculpt brain synaptic connections. And notable research accomplishments include the discovery that microglia, which is one of the immune cells in the brain, prune neural connections in response to signals from the classical complement pathway, which is one of the immune signaling systems in the body. Additionally, her lab has uncovered a diverse set of other additional immune molecules that regulate synaptic modifications in the brain, both during normal development and also more recently in Alzheimer's disease, which she will talk about today. Her work uses interdisciplinary approaches and straddles fields of genetics, immunology, and neuroscience, as you will come to see in her lecture. Today, she will present cutting-edge work on the role of microglia in the pathogenesis of Alzheimer's disease, and we are very, very grateful that you have agreed to give this lecture today. So Dr. Stevens, take it away.
Thank you so much. I'm so happy to be here today. Thank you so much, Dr. Messing, for that introduction and the opportunity to come today and speak and share some of our work with you all. So today I'm going to talk to you about microglia, our resident immune cells or macrophages that live in our brains. And these cells really have emerged as central players in brain development and function, but compared to other cell types in the brain, we still know remarkably little about their biology and their normal functions, which is really key if we're to understand how they go awry in diseases like Alzheimer's disease. So as you heard, we discovered that one of their many physiological normal functions is to remodel or prune synapses, which in the developing brain is a normal process by which our circuits get sculpted. But the more we learn, the more we're really appreciating that pruning is really just the tip of the iceberg in terms of the functions that these cells have in the brain. And so today I want to share with you a little bit about the background of microglia and some new work that we in the lab have been doing to try to uncover how they signal to other cell types in the brains and potentially even the body, and how environmental or genetic factors can actually influence and lead to microglia dysfunction. So one of the things we certainly appreciate, and this is thanks to the many tools that have been developed in the field over the last many years, is that these cells, which are resident myeloid cells or macrophages, really they tile the brain. They make up about 6 to 10% of the cells in our brain, but they're remarkably responsive to changes in the environment. And this is really beautifully illustrated in this video, where you can see live imaging within the brain of a mouse where the microglia have been labeled in green. And this is just recapitulating one of the classic experiments that was done by Wenbiao Gan's lab and Axel Nimmeron years ago, where you can see a laser making an injury in the brain, and all those microglia processes just gravitating and migrating towards the site of injury. And this is something that is very robust, and what it raises a lot of questions, right, in terms of what are they recognizing? What are the signals that are bringing microglia to that site of injury? And once they get there, what are they doing? One of the things we know that they do, because also our brain's resident phagocytes, is that they can really rapidly clear or remove debris like that's been generated by this injury. But we also know that if the brain environment is such that it's not really enabling them to go back to their homeostatic roles, this can lead to the production of neuroinflammatory molecules, and ultimately you can get neuroinflammation. This is just one of the many functions of microglia, but I think this beautifully illustrates what microglia can do in the context of an injury. But of course, this term microglia activation, which I'm going to use in quotes here, is a term that we and many in the field have used to describe a change in microglia, a change in their morphology from this sort of really beautiful process-bearing cell type to one that's quite different, right? It's pulled its processes in. These cells start to express phagocytic receptors. They become more lysosomal activation or activity. And so we know they're different. And oftentimes when you see a microglia that changes state like this or function, or morphology, we see it in a snapshot from either fixed brain tissue from a human brain or from a mouse model, for example. But what we don't know when we see that these cells have changed in a snapshot image is when did they change? And most importantly, how do microglia contribute to specific aspects of disease pathogenesis? And this is really the question that lays before us, is to try to understand the when and the how. And if we can understand both of those questions, especially the how, that might enable us to develop new therapeutic targets and new therapeutic strategies to try to really push them into the more beneficial state and decrease their detrimental functions in the brain. Now, what we now appreciate is that, although a small fraction of Alzheimer's is late onset, the early onset Alzheimer's disease, we now appreciate that microglia, a lot of the genetics are implicating myeloid cells in microglia. So these cells were long thought to sort of become activated and dysfunctional, sort of secondary to neurons, but emerging human genetics and GWAS studies more and more are implicating genes and variants that are expressed or enriched in microglia and myeloid cells. And these are just illustrated here in blue. This is a schematic or a figure that was modified from some of Alison Goet's work that just shows that a lot of these genetic signals in blue, we now even have gene names. And we now know from expression profiling that many of these genes, including TREM2, CD33, and even ApoE, are expressed and in some cases enriched in microglia. So that really has changed the game and the landscape in terms of thinking about microglia, and because the genetics are telling us these cells are critically important. But again, how do we go from the genetic findings like this to try to understand what functions and what mechanisms are going awry, and how does that relate to cognitive and memory impairment in Alzheimer's disease patients? So the challenge with trying to connect the dots between these genetic leads, which is incredibly exciting and important, to specific functions, is that microglia, because they're our resident immune cells and macrophages, they are incredibly dynamic and they exist in lots of diverse states in the brain. And so I've sort of illustrated a couple examples where they're normally sort of tiling our brain and surveying the environment. They can also, however, play lots of different roles depending on the timing and the context. So for example, in development, they play critical roles not only in pruning synapses, as I'll tell you about in a moment, but they're important in clearing dying newborn neurons. They're important in sculpting circuits. And they're also now known to be important in modulating neuronal and synaptic plasticity. So this is work now been shown by many in the field. So we know they have a lot of these normal functions that are important for normal, healthy brains and synaptic circuits. But when there's a challenge, whether that's environmental or genetic, these cells can dramatically change states, as I showed you in that earlier movie. The problem is we don't at the moment have a deep understanding of what the triggers are that convert this sort of brain surveying homeostatic healthy microglia to one that's now more inflammatory. Or, for example, a microglia that can suddenly become more phagocytic. And so, again, there's beneficial and detrimental functions of microglia. And now the challenge is to try to better understand what the signals are that convert them to these different states and develop new ways to identify markers that denote these different beneficial and detrimental states. So today in the first part of my talk, I'd like to tell you about what we've learned about one important function of microglia and their role in synaptic pruning, and some of the molecules and mechanisms that we and others have uncovered that allow and enable microglia to prune specific synaptic connections during development, and how understanding that is also providing insight into pathological synapse loss, which is a hallmark of Alzheimer's disease and other disorders. So we know that microglia is a key component of neural circuits. So if you look at this beautiful image of a rainbow mouse from Jeff Lichtman's lab, most of the time you're sort of honing in on all of these different populations of neurons shown in these different colors. But if you really zoom in, of course, the brain contains billions and trillions of synaptic connections, the connections between neurons shown here in purple. And if you then overlay microglia, we can appreciate that these cells are really intermingling and interacting with these synapses, these puncta here in pink. And this caught our eye a number of years ago when I started the lab. And this is work that I'm going to share with you by Dory Schaefer, a postdoc in my lab. We both became really interested in understanding, could microglia be dynamically surveying and interacting with synapses? And could they be sculpting these circuits? And if so, how? And this is just another movie to show an example of a microglial cell in the cortex of a mouse. All the microglia are in green. This is just one branch of a dendrite, and these are the synapses. And again, we observed that these microglia are constantly touching and interacting with synapses. So there's subcellular interactions that we're really interested in understanding more deeply. So before I showed you that when you do something really dramatic, like damage the brain, microglia really move in towards the site of injury. But what we were really excited about is the idea that even local signaling between neurons and synapses can release molecules that can then recruit or change microglia dynamics and functions. So what could some of those molecules be? Now, during development, microglia are, particularly phagocytic, meaning they have a lot of phagocytic receptors. We were noting that during these critical periods in development, when there was a lot of remodeling that was going on, microglia expressed a lot of these phagocytic markers. So putting the two ideas together, those videos and those movies, with some of the observations of microglia morphology and phagocytic receptor expression, led to wonder whether they were literally nibbling off some of those extra synapses in the same way that a circulating macrophage or a tissue-derived macrophage in the body might model or prune away or engulf a pathogen or a piece of debris. And so we wondered, could they use some of the same biology, some of the same machinery that macrophages and other parts of the body use to clear? So this is the circuit that we've been studying. This is the visual system of a mouse. And it's a circuit that's been beautifully characterized and well described at the functional level, at the anatomical level, and at the electrophysiological level by many in the field, including Carla Schatz and Chin-Fei Chen and many of my colleagues at Harvard. So we decided to use and build on this model system to try to understand more deeply whether microglia prune synapses, and if so, is there any specificity in the process? And if so, what are the molecular instructive signals? So this is the circuit we're talking about. It's the retina and the visual thalamus. And fortunately, you can actually put dye in the eyes of mice and label those retinal ganglion cells with two different colored dyes that then trace the circuit into the brain. And so here I'm just showing you a zoom in of the visual thalamus, the dorsolateral geniculate nucleus. It's the visual thalamus where you can see eye-specific projections that go to very specific regions of the visual thalamus. And shown over to the right is just an example of a neuron here, a postsynaptic neuron, that's innervated by lots of these axons from both eyes early on in development. And then over this critical period of 2 weeks in a mouse, you see the pruning away of the blue inputs from one eye and the strengthening and maintenance of the red inputs. This is a term that's been coined by Carla Schatz, essentially use it or lose it, right? Where you sort of, you keep the connections that are meaningful, in this case, the red ones, and you prune away the ones that are less meaningful. And we know that this is regulated by neuronal activity. The idea that more active synapses, in this case, the red ones, that are able to fire the postsynaptic cell and communicate with it, tend to be the ones that stay at the expense of their less active blue neighbors. And so we wondered, could microglia be reading out these activity-dependent cues? Could they be preferentially engulfing the less active inputs, in this case, the blue ones? And this was such a beautiful model to study that. And so when Dory Schaefer was a postdoc in my lab, she built on this model and asked that very question. Do microglia engulf or nibble off those synapses in the normal healthy brain? And if so, are they doing it in an activity-dependent manner? And this is just one example of a microglia intermingled with all those little synaptic inputs in red and blue. That's like a zoom in of this box. And what we found is that almost every microglia we surveyed during this window of development were remodeling or engulfing these synapses. We could even see it at the electron microscopy level. And what we then asked is, what are the cues? And what we found is that microglia were actually recognizing a group of molecules that had been long studied by immunologists, a group of innate immune molecules called complement. These are secreted immune molecules that normally are our first line of defense against a pathogen or a bacterial cell in the body. And what we found are these molecules also are expressed in the healthy, normal, developing brain, and that they are essentially binding to subsets of these synapses, just like shown here, and that microglia have receptors that recognize these molecules. And long story short, we went on to show that if you block either the complement molecules or the receptors on the microglia, that led to pruning defects. So that it basically, these microglia could no longer recognize and prune or engulf these synapses. And that told us this was one important pathway that was important in synaptic pruning. And we've gone on to show and really delve deeper into the mechanisms, which I won't go into here. But I just wanted to give you a sense of how we can learn from what immunologists have been studying in the body and apply some of this biology to try to understand a really complex question about synaptic specificity. And the idea is that complement is targeting subsets of synapses recognized by microglia. We've also uncovered another group of molecules that prevent or protect certain synapses from being eliminated. These are called don't eat me molecules. And the model is that when microglia, by surveying the brain, if they see a combination of cues on a synapse, like in this case, have the don't eat me signal and complement, even if complement's on those synapses, it will not engulf the synapses that have the protective molecule, but they will engulf this combination. And so we think just like the immune system, the brain and the microglia, based on their receptor ligand combinations, can read out different cues on synapses, and that's telling them which ones to prune and which ones not. And now this is very tightly regulated in the healthy brain. There are lots of ways to make sure this doesn't go awry. And so we started to wonder, maybe what's happening in the context of Alzheimer's and other diseases of the aged brain is that some of these control mechanisms go awry, and now you get an aberrant activation of this mechanism of pruning. And indeed, synapse loss is a hallmark of many brain diseases, including Alzheimer's disease, but also neurodevelopmental and neuropsychiatric disorders that raise the question of whether there could be a common mechanism downstream that renders synapses vulnerable in disease. And indeed, we've gone on over the last many years, mostly using animal models from different disease contexts, to show and provide evidence that supports the idea that there's a reactivation of a developmental pruning mechanism that involves this neural immune microglia synapse interactions, and in particular involving the complement system. And so I just wanted to highlight that really briefly in the context of Alzheimer's disease. We can start to ask, well, okay, are these complement molecules dysregulated in Alzheimer's disease patients and animal models? Are microglia aberrantly pruning synapses? And of course, these are the questions that we have been gone on in parallel to our developmental work to try to test. And as I mentioned, synapse loss is both an early event in Alzheimer's disease, and it's the strongest correlate of cognitive decline. And so we wondered, again, much of the field, of course, had been studying microglia in the context of neuroinflammation and plaque and pathological hallmarks. We wanted to know, could microglia be involved in a very early stage of Alzheimer's disease when there's early synaptic dysfunction? In this sort of, in the patient's case, this would be a preclinical Alzheimer's. And in animal models, we have the unique advantage to be able to take existing models, rewind the clock to the beginning before there's plaques and before there's pathological hallmarks, and start to see what's going on with the microglia and these immune-related pathways at the very earliest stages of disease progression. And then if we understand the mechanisms in the animal models, could we then try to validate those models and mechanisms in humans? And that's exactly what we've been doing. As our first proof of concept, we started to look in some of the animal models that do exist for Alzheimer's disease. None of them are perfect, but they are a good starting point to test the hypothesis. Does complement-mediated pruning contribute to synaptic dysfunction and loss in Alzheimer's disease? And what we first observed, and this is work by So-Yeon Hong, who's now faculty at University College London, and she was trained with Dennis Selko, and she was the first really in my lab to really apply some of her expertise in Alzheimer's disease and bring that to my laboratory. And what she observed in 2 or now 3 different animal models of Alzheimer's disease, we could see complement, remember C1q, really brightly depositing on synapses and in these brain regions like the hippocampus and frontal cortex, very, very early across this disease progression in these animal models. And also there was region-specific vulnerability, meaning we were seeing a lot of complement in regions like the hippocampus involved in memory or frontal cortex versus other brain regions. And when we zoomed in on that, we could see, just like development, these complement molecules were binding or tagging subsets of these synapses, except now instead of it being a good thing, necessary for normal synaptic wiring, now we thought this might be pathological. And indeed, we showed that if we knocked out a lot of the different elements of the pathway, C1q, complement, or the receptor on microglia, we could actually protect and rescue not only the synapse loss, but some of the memory and cognitive defects in these animal models. And indeed, microglia were overeating or over pruning synapses in this model. And so this, again, it was a kind of proof of concept early days. This was published a few years back, but now other labs have gone on to show similar findings in other animal models, including a tau model of Alzheimer's. And now we're in the position of starting to ask, could we actually start to translate this into humans? And we're doing this in a couple of different ways. I first just wanted to highlight that there's a C1q blocking antibody that was developed by Annexon Biosciences, which was a company that was founded by my previous mentor, Ben Barris. But essentially, this is an antibody that's now in the clinics, but we used it as a tool compound and showed that when we treat mice with a C1q blocking antibody, we could also protect the synapse loss in these animal models, and others are going on to show similar results in other animal models of disease. So this is quite exciting because this is starting to go into the clinics now, not in Alzheimer's disease yet, but as a proof of concept to show that this could be protective in diseases that involve synaptic loss. And we also, I think, important to note that one of the challenges for trying to think about therapeutics is that, you have to really think about context and timing. Because I told you today that we've identified an important role for complement in synaptic loss, which we think is a detrimental function of complement. And if you block it, it's protective. But if you wait too long, you don't want to disrupt all the beneficial functions of microglia and complement in the brain. For example, we know that both complement and microglia are really important in clearing plaques and also preventing other aspects of clearing dead cells. So we want to think about when and where to intervene, which is why we really need to dig deeper into the mechanisms by which this works. So this really kind of begs the next big question is, can we start to come up with better markers to both define and I would say redefine microglia states and functions so that we could non-invasively, for example, start to track some of these immune molecules and immune cells, in this case microglia, across disease progression longitudinally. And if we could do that, we could kind of hone in on the window in which to start to really target, and to also maybe even start to think about how to stratify patients if we had biomarkers, for example. So how do we do this? And this is really a challenge, because I only told you in my talk thus far, really, that we've been focusing in on one major function, which is synaptic pruning, but microglia do a lot of other things too. Could we develop, for example, new markers that actually denote and actually enable us to track specific disease-associated populations or states of microglia, and then that would enable us to try to target more specifically the detrimental functions of microglia versus the best beneficial. And over the last several years, I think, again, new tools and technologies has really opened up the field and given us new strategies to try to tackle this question, including single-cell sequencing, where you can finally, for the first time, take a piece of brain tissue from either human or animal models, dissociate into individual cells, and thanks to this new technology developed by Steve McCarroll's lab and Aviv Regev and Evan McCosco, now we have routinely ways to single-cell sequence all the cells in the brain, including microglia, to try to better understand diversity. And that's generating these sort of transcriptional maps that give us genes that actually denote these different clusters or states of microglia that are actually providing us new insight into the different populations that change across development, aging, and injury and disease. And from that, we are now able to start to think about how we can identify the most relevant disease-associated populations. And the first group to really use this technique to try to identify disease-associated microglia and Alzheimer's was Ido Amit's group, which used that approach to take animal models of Alzheimer's and again, characterize the microglia states. And they have identified a disease-associated microglia state called DAM that now many, including our group, have reproduced. And they show that a lot of these disease-associated microglia, which express genes related to lysosomal function, phagocytic activation, they are often found around the plaques. So that's giving us some clue. But I think it's also kind of oversimplifies to some extent, because we still don't know, are they detrimental, beneficial, and are there other relevant macrophage or myeloid populations we should be studying? And that's really where I just want to lay out a couple of the big questions I think the field is now needing to tackle. We need to better understand how dynamic these states are. Can we reverse them and bring them back to a beneficial state? What are the functions of all these different populations that are coming out of the single cell data? And most importantly and relevant, how do different genetic and environmental challenges alter these states and functions? And so I think many are starting to tackle these questions in the field using really collaborative and integrative approaches. And we also want to start to think about how we can better faithfully model human microglia, because I just don't think animal models are going to do it. We need to come up with new ways to do that. And this is really in the last couple of minutes, I just want to highlight work that's now ongoing in the field, thanks to some really pioneering work by Matt Thurton-Jones, who's going to join us in a moment, where we actually can start to model microglia in humans using human IPS models. And collaborative work now at the Broad, in other collaborations in a consortia that involves Matt and Allison, we are starting to develop ways to start to study microglia in vitro and ask the question, how do different challenges, both environmental and genetic, alter microglia states and function? If we could grow them in a dish and try to recapitulate what we see in a human brain, that would enable us to develop pipelines in a more tractable system to really deeply systematically identify mechanisms that can then be validated in vivo. And so we wondered, could we actually use single-cell sequencing to start to ask about diversity and functions in these microglia IPS cells? And indeed, what we're finding, to our surprise, is they do exist in multiple states. This is another one of those TSNE plots that just show all the different transcriptional signatures, much like I showed you earlier. And indeed, even in a dish, we can observe a lot of these different states that are very quite interesting and relevant, including this DAM, or this disease-associated microglia that I just told you about. We can actually see these microglia in our in vitro models. And we can then ask, are these relevant to human? Because work that we're doing in collaboration with Evan McCosco and others, we can actually take single cell data from human patients and then integrate them with our single cell data from the IMGLs or the IPS work. And indeed, we see multiple populations, these chemokine populations, proliferating populations, and even those DAM populations. So this is quite exciting. It's all unpublished work, but I just wanted to highlight that we actually think we do have the ability to start to ask questions about what triggers disease-associated microglia? Are they good? Are they bad? And they are all also being stimulated by very specific stimulus. So we can start to really tease out what those triggers are. And we can then start to ask, how do we bring together the genetic risk to specific functions? How do we link the genetics to specific functions? And that's ultimately what we're going to need to do to get closer to a more specific translational approach to get at new targets. And we can even start to do functional assays. Like I showed you pruning in vivo in the mouse, we can also do pruning or engulfment in these in vitro models. And we can use different patient-derived microglia lines or different lines that lack the different genetic variants and ask how that alters things like phagocytosis. And this is just one movie to show microglia in a dish. You can see some of them are engulfing a lot of synapses or a lot of amyloid, and some of them are not. Again, giving us the ability to start to identify what makes these cells different from one another to try to come up with new targets and new approaches. And even as a proof of concept, we can knock out a gene like TREM2, which has been shown to be a risk gene, and we can show that influences microglia disease-associated signatures, but also their ability to phagocytose, which has also been shown in animal models. So this is a nice proof of concept. So, and I just want to say very briefly that work, again, by Maplert and Jones is now enabling us in the field to start to be able to go back in vivo. He's developed these really beautiful chimeric models where you can essentially transplant these human microglia into a mouse brain. And you can see this beautiful chimerism that he's observed and developed protocols to be able to study. So we can imagine taking microglia from a patient, studying them deeply in vitro, and then selecting lines to inject into a mouse to then study the consequences. And I think in the end, using a really integrative and collaborative approach, there's many different ways to be able to tackle hard questions. We want to be able to really map and connect the dots between the genetics and these challenges with specific biological pathways so that ultimately we can start to target very specific functions of these cells. And I think this could identify not only new therapeutic targets, but hopefully new biomarkers, including new imaging biomarkers that might enable us to start to track microglia non-invasively in patients across time. And I think this would be really exciting. And I think that's where some of the applications of the single cell data and these functional studies are taking us. So I'll end there by just thanking the microglia, human microglia in my lab. This is just a fun photo. Lots of amazing collaborators and support. So thank you very much.
Beth, thank you. Thank you for that beautiful lesson on microglia. And to everybody, I'm Bill Schwartz. I'm professor of neurology and integrated biology here at Dell Medical School, the University of Texas at Austin. And for the rest of the program, we've invited two discussants who Dr. Stevens has already introduced to you. From the left coast is Dr. Matt Blurton-Jones. Matt is an associate professor in Department of Neurobiology and Behavior at UC Irvine. He's also the director of the stem cell core there. And as you've just begun to hear, his research is powerfully exploiting induced pluripotent stem cells and mouse models to elucidate the molecular mechanisms, including those in microglia that might drive Alzheimer's disease. And from the right coast, we've got Allison Goetz. Allison is a professor of neuroscience and chair of the Department of Genetics and Genomic Sciences at Icahn School of Medicine at Mount Sinai. And you'll know, of course, that since the 1980s, Allison has been elucidating the genetics of neurodegenerative diseases, including the enrichment of Alzheimer risk variants in the regulatory elements of microglial DNA. So we're glad to have both of you on the screen, and to the audience as Matt and Allison get going. Please submit your questions through the Ask a Question button, and we'll hope to get to as many of those as possible. Allison, do you want to lead us off
Sure, happy to. So, Beth, you really nicely demonstrated that microglia exist in many different states. And we know that those states change during disease. I wonder whether you could comment about which states do we think are likely to be protective versus detrimental, and if we're thinking about therapeutics, what do we want to do? How do we want to affect these microglia in order to be beneficial?
Yeah, I think it's a great question, Alice, and I think this is really where many of us are starting to try to head, and the first step was some basic characterization so we can get a way to even identify that there are different states. And I think we're past that now. I think we have a pretty good handle on the fact that there are now markers, at least based on transcriptomics, that can kind of give us a little bit of a foothold into those different populations like I talked about today. But I still think it's overly simplified because there's a couple things we still don't know. One is even the disease-associated microglia, that DAM population, that can very well be both beneficial or detrimental depending on the context. And I think that's still what we're grappling with. I suspect there's lots of flavors in between, lots of shades of states in between that we haven't even come to yet. And I think one of the things we need to do now is start to be able to really more deeply characterize how dynamic are these states. Like, we don't even know, for example, if a microglia that's homeostatic, kind of surveying the brain, doing all the good things it does, if it shifts to a disease-associated signature, is it stuck there or can it go back? Like, are we able to, are they plastic, right? And that's been really hard to study in vivo because we haven't been able to we get snapshots. But that's one of the things I'm really excited about with some of the IPS models that you and Matt and others are starting to use, because we might be able to time lapse and image this in real time with reporters and ways to study that. And then if we could do that, we could link that to specific functions. So we still have a bit of work to do there, but I do think we're kind of right on the edge of the next step of trying to understand that functional aspect, which can only be done with new tools. So I think we still need more tools.
Yeah, so have you or Matt done single cell sequencing in culture dishes of induced microglia to know how much of the variability that we can see in a living brain are we also able to see in a tissue culture dish? So how much are we going to be able to rely on in vitro models versus what Matt is proposing, obviously putting human cells into a mouse brain, which is very elegant, but also more time consuming to do. And so,
I mean, I will just say something briefly based on a slide I showed at the end, which is the first example of whether we can more deeply validate the IPS model that Matt's developed. I'm sure Matt can say more on this because we're actually collaborating, Matt and I, on a couple of these projects, because the idea is that, like, do those different states that I showed you today by single cell, how close are they to the human brain? And this was, again, thanks to collaborative work where Evan McCosco's group has been taking human frontal cortex of individuals that do and don't have amyloid, tau, or Alzheimer's pathology. And what he's showing is this diverse signatures from the human brain. And then from all of that single cell data from the human brain in vivo, we then integrated that with our IPS modeling, and it was almost completely aligned. Which means the states that I showed you, all those different colors, were the same states that we're seeing in the human patient. So that gives us some hope that the states that we saw are relevant. So now the question is, like, how do we push that to the next level and study function? And there's only so much you can do in vitro. Then you've got to take it back in vivo. And I think that's where, Matt, you can comment on how you can then build on the chimeric models to get closer to what are the functional consequences? How does this affect cognition? How does this affect memory? You can't do that in a dish, right?
Right. No, thank you, Beth. So let me start by saying fantastic talk, as always, really informative. So yeah, in terms of the chimeric models, I mean, these are, as you introduced them, this is the concept that we could take human stem cell derived microglia and be able to put them back into a specialized mice and be able to see how these cells function in vivo, in the brain, how they interact with pathologies like the amyloid plaques that occur in Alzheimer's disease. And so we're just getting started on that, but so far it seems to be quite informative as a new way to model the function of these cells. as with every experiment, there's caveats to this. So we are still looking at human microglia now interacting with mouse neurons and mouse astrocytes. And so obviously there's still a need to really understand what's happening in human brains. And I think, Beth, I know you've been sort of leading the charge on this. And I think it's actually important to sort of acknowledge the challenges that we face with studying human microglia from human brains. I wonder if you might want to comment a little bit more about where that data came from, because it's really interesting project that you guys are working on.
Oh yeah, thanks, Matt. Yeah, in the interest of time, I didn't mention the source of the tissue, but it also brings another question up, maybe for a group discussion, which is like we are now able to go into post-mortem brains, and in the same way, Amit's first paper showed you can do in a mouse model of Alzheimer's. Many in the field are starting to take post-mortem brain that are banked in brain banks and do single cell and see all kinds of interesting, signatures and states, including DAM, right? Great. The question is like, how relevant is that? Like right out of there's going to be some artifacts and some issues that happen with post-mortem brain. How close is it to human? So we have a collaboration with a neurosurgeon up in Finland. This is a large collaborative effort with Evan McCosco's group and a neurosurgeon who's been collecting surgical samples from patients that have idiopathic normal hydrocephalus, this INPH. Now, these aren't Alzheimer's patients, but quite interestingly, they're between 65 and 90 years of age, and many of them do have cognitive and clinical symptoms of AD, kind of preclinical AD, and about 30 to 40% of them have either amyloid or tau pathology. But the power of this is that this neurosurgeon for many years has been going into the frontal cortex, releasing pressure, collecting CSF, which is normally what you do, but also banking all the brain samples that came out of the pipette. And that's the data I showed you today, was that punch of tissue, which was right out of the head into a freezer, no delay. And that's what gave us that high quality data. So I think in many ways, it's a great kind of proof of concept where you can go into very fresh tissue. Normally you can't go into people's brains and sample whenever you want, but this is a unique cohort who's still living, that not only do we have the brain, but we also have CSF, blood, and a lot of information on these patients that you can start to then map out the differences between the brain states, the blood, CSF, and ultimately clinical, cognitive, and other sort of important like genetic data. So the genetics will be really interesting there. Also, if you can start to think about some of the work Allison's doing to understand ApoE and other genetic risks, and how does that influence some of these states in these particular patients?
So we have a number of questions from the audience that are pouring in, I think, for sort of all three of you. So let me see if we can tackle some of those. First question from, or is the question, does C1q coat all healthy neurons or only those that are not signaled during developmental pruning? If the latter, how is that regulated by signaling?
Yep, thanks for excellent question. The interesting observation I made even back when I was a postdoc with Ben Barris was the fact that not all synapses get tagged with C1q. There's subsets of synapses, and after a certain developmental period, we don't see a lot of tagging anymore. So even within a region, there's subsets of synapses. So what we're finding is that there must be molecules on the surface of some synapses that are recruiting complement and microglia to those synapses, and a lot of those are immune related. And that brings us to this question of like, what's the signal? And we think it's some of the same like membrane surface receptors and proteins in development are also being exposed in Alzheimer's and in disease. So if we could understand what those molecules are, that might be the target, right? How to approach it from the synaptic level. And one of the things we recently identified, is a paper we published this past year, was phosphatidylserine, which normally in a healthy cell is in the inner leaflet of a cell, but we showed there's a flipping of this lipid phosphatidylserine to the outside, and that recruits complement and microglia to it. So that's just one example of like a signal that could give specificity. Because in the same way we see in development that there seems to be specificity, in the Alzheimer's models, we're also seeing vulnerable brain regions and vulnerable synapses tend to be the ones that have complement on them. So I think understanding that mechanism is going to be really both interesting, but also maybe therapeutically relevant. If we could find, like, for example, a small molecule that would target the binding proteins versus complement itself.
Yeah, I think it's probably important to point out there, Beth, too, right, that the phosphatidylserine is actually a ligand of TREM2 on microglia, and this is one of the more recently discovered genetic risk factors that's really focused everyone's attention on microglia because of TREM2's role in microglial function. So
right. Yeah, so I guess the model there, and we have a little bit of data on this too, that TREM2 might then be like the receptor that recognizes phosphatidylserine on the synapse, because it's not all about complement. Complement's just one way, but that's a really interesting mechanism that we could think about testing in the context of AD. Another link. Yeah, thanks.
Have you? Here's a second one, again about mechanisms. Have you all looked into how the production of reactive oxygen species as a result of the microglial activation might affect disease progression?
Anybody want to take this one? We haven't been doing any work in this particularly.
Yeah, I haven't done a lot, but it's well established that microglia, when they get activated in certain ways, they'll produce nitric oxide and other reactive oxygen species that certainly damage other cell types, neurons. I think it's one of these things that we're going to learn a lot more about as we start to define these states, because we know some of the genes that will regulate either the production or the ability to handle some of these reaction oxygen species. And as we see when those genes are getting turned on or off in proximity to different pathologies, I think we'll learn a lot more. At least initially in terms of Alzheimer's, it looks like that pro-inflammatory oxidative state there's some evidence that it may increase some of the tangle pathology, the tau pathology that occurs later in the disease. So it may be sort of an example of a negative consequence of certain types of activation.
Another question has brought up the issue that you raised, Beth, about rodent versus human models. Could you discuss a bit more about how rodent dams differ from human dams, and how the ligand receptor interactions in the chimeric models might actually recapitulate human disease?
Yeah, great. I'm sure all three of us might have something to add to that. I think it's still early days, but based on some of the data that's been analyzed thus far in the field and also in our own groups, there's definitely some conserved DAM signature that we see across human and mouse. And that's really interesting because you can, at least fundamentally, there may be some core component or core mechanism that seems conserved. That said, when you start to really look in human tissue, especially that high quality fresh tissue I mentioned, where there's a lot of microglia, and the more microglia, by the way, you have, the more diversity and things you might see, because a lot of these states exist in very small populations. So if you don't have the power to pick it up, you'll miss it. So sometimes when you see a difference, when someone compares mouse and human, for example, and they say, oh, actually, this is not in human or this is not in mouse, it might just be technical because you don't have the power to pick out all the cells. That said, I'm sure there are going to be differences. And actually, there have already been some comparisons between mouse and human and other species that are already quite interesting. And maybe Matt and Allison might want to comment more on that.
Yeah, I mean, so that's a great summary. And part of the reason we really started to pursue iPS-derived human microglial models and then chimeric models are that some of the genes that Allison and others have really highlighted have some big differences between the mouse version and the human version. In fact, some of them don't have an appropriate homologue. And so there is this need to study the human cells. We at least know from the chimeric models that there's a partial overlap, as Beth alluded to, in the DAM signature. Some of those really strong DAM genes are still there in the human version of the DAMs. But when we look with the studies that are coming out from single cell sequencing in humans, again, it also suggests only a partial overlap. And I think there's potentially a number of explanations. One is quite simply the DAM signature is defined in mice has so far been from amyloid-only models, so models that just produce beta-amyloid pathology. And we're comparing them to Alzheimer's patients who have both amyloid and tangle pathology and neuronal death. And so there may be different triggers. And in fact, we have some new data looking at a signature in a tau mouse that looks very different. It's a different type of activation state, so more of an interferon activation state that one sees. So the DAM as initially defined may be a more amyloid-dependent state, and in humans you have more of a mixed bag from the different
Right.
I think perhaps one other thing to add here is that, microglia are, the macrophages of the brain. And if we look at macrophages in other tissues in the body and responding to similar lipid-rich debris, that there's a core signature in those macrophages that definitely overlaps, is similar with the DAM state that you see in microglia. So some of this response we're seeing in the brain is really probably part of a core signature response of macrophages in different tissues.
Yeah, absolutely.
Yeah. Just a quick note, just to bring in something I quickly touched on at the end, is this idea of like, it could be that different triggers, different challenges induce these different states. And that's some of the in vitro, that's one of the reasons why we set up the in vitro assay, where you could say, okay, if I give apoptotic neurons versus amyloid versus synapses, like, do the microglia care, or is there a preferential, response to the different challenges? And we are seeing differences in the different challenges. So that to me also suggests that could be different between the model, right? Depending on what the brain environment has in it at that time. So I think that's it's also much to be done there, but it suggests it's not an all or nothing response, right?
And that highlights regional differences too, right?
Yes.
You get a different response in the white matter versus the gray matter, or versus the hippocampus versus the cortex as well.
Yeah, and it kind of emphasizes also our inadequacies or limitations with our animal models, right? That we don't have an animal model that really looks like Alzheimer's disease.
Right.
We have tau models and we have amyloid models, but not Alzheimer models, unfortunately.
Yeah, exactly.
So getting back to development a bit, are the DAM cells able to detect the "do not eat me" signals, such as those found during development? And if so, could manipulating expression of those signals be sort of another input, an avenue to reduce disease progression?
That's a great question. We've been thinking a little bit about that with the context of the CD47 I mentioned very briefly. That's a very classic "don't eat me" signal. Irv Weissman's group and many others have been studying this in the context of cancer and as a therapy, right? So immunotherapy, because it's a way of basically tricking the macrophages to get rid of a tumor cell. So the idea that maybe some of these molecules in the brain could also be interacting with disease-associated microglia, we could easily do that experiment. I guess we could take the CD47 knockouts and see how that impacts, disease-associated microglia or microglia function in the context of Alzheimer's. We haven't done it, but it's certainly a very interesting question. And it gets back to this idea of, there's many ways to target this. You can either go after the detrimental part, or you could try to increase the protective or beneficial. And I think, Allison, you asked that question at the beginning. There's lots of ways you can imagine, like even in the context of ApoE, where you have protective alleles and obviously ones that are at higher risk. How do we leverage that to try to understand, maybe not only to go after the negative, but how to boost the positive, right?
Absolutely. And I think, quite a bit of research in the genetic side on Alzheimer's disease is focused on trying to understand protective mechanisms, because it really identifies, right, nature's protective mechanisms. If we can mimic them, we should be able to develop therapies. And there certainly are several companies that are focused on ApoE2 as a protective mechanism. And is there a way to mimic that? I think we've also seen in the last couple of years, in the Colombian kindreds with presenilin mutations, there are people who are outliers, live until their 60s and 70s with no cognitive impairment. They have a brain full of amyloid, and yet they didn't develop disease. And so that does show you that if you can prevent the degenerative phase of the disease, you could actually live with a lot of amyloid in your brain.
Yeah. So that's good news, right?
Yes.
So we're reaching the end of the hour, and there's a lot of excitement with questions, which we won't get to all of them. So since we can I ask a quick question? You know, when we talk about synaptic remodeling in physiological states, development, of course, I also think about sleep and synaptic homeostasis. Do we know anything about
You're asking my favorite question.
how are the microglia behaving during sleep? Are we missing the most important cell type during sleep?
We're studying this very question. Someday, when maybe a year from now, I'll come visit. Hopefully we can show some data on this. But yeah, this concept of like, could sleep or circadian changes, could that influence pruning? And there's been some data to support that when you sleep, you're remodeling a lot of the synapses and you're also clearing waste. So one idea is that that's one of the reasons why we sleep, is to make sure that happens. This is a homeostatic mechanism. And that maybe it might also kind of bring together the immune component with some of the synaptic components along these lines. So we're trying to set up some experiments now to test that hypothesis by manipulating sleep and circadian and see how it influences things like pruning, and then how that also changes in the context of like sleep versus wake. So lots to do there, but I think it's a really interesting idea.
Last question. Do you see a pathway for a cure?
Go ahead, guys.
Well, I think we have to be more optimistic, at least, than that I think we know a lot more about the pathogenesis of Alzheimer's than we did 20, 30 years ago. It's obviously frustrating that the amyloid therapies in themselves have not produced the results in terms of treatment. But I think the genetics in the last few years have pointed us all in a new direction, right? I mean, none of us were really studying microglial biology in the context of Alzheimer's disease. And so I hope that there will be some new therapeutic targets that come out of the kinds of studies that Beth is doing. So.
I'd second that. I mean, I think we're in a renaissance based on Allison and other geneticists' work who've identified these microglial genes. We have new targets. Up until now, pretty much every therapeutic approach has gone after amyloid, but we know amyloid accumulates for 20 or some years before people develop symptoms. So that's a very hard thing to target. Now we have something a little bit later that seems to be very important. Microglial function, probably influencing synapse loss, which is ultimately what correlates best with cognitive impairment. So I think we've got brand new targets, and the more we learn, the more we're going to be able to focus in on more precise therapeutics.
I know we only have 1 minute left, but just to echo both of what you both just said, just to say one last piece, which is I think we are a lot closer, and I think it's a really exciting time in the field. Not only do we have new tools and approaches to tackle this, there's a lot more collaboration in the field that's never been there before. I think it's exciting to bring in people from different disciplines to work together on a really hard problem. So that's key, sharing data, sharing our knowledge and expertise with each other. But I do think it's probably not going to be one magic pill or one way, that if you can understand microglia and try to target them, that would be amazing. I think we won't be that far from thinking about ways to do that. But it still starts in many cases in the neuron. So something's going on to make those synapses and neurons vulnerable. And I think it's critical we understand that too. And then ultimately bring them together, right? So studying them both together, I think, is going to be key, right?
Great final words. Thanks to all of you, Beth, Allison, Matt, for this introduction and lesson on microglia. And with that, we turn it back over to Bob Messing.
Yeah, I think so. Thanks, Bill.
I will try and take just 1 minute here to thank you all for taking the time to do this presentation today. Beth, for your wonderful lecture, and Matt and Allison for joining in with questions and adding a great deal of information about your areas of expertise. And I would agree with you. This is a new area to tap into, and it's a very exciting area in which we're learning a lot more how to manipulate these neuroimmune systems, and hopefully for the benefit of a cure. I would like to invite everybody to our next lecture, which will be scheduled for September 23rd, yes. And it will be given by Mark Diamond, who's at UT Southwestern. Mark is an expert in tau-related disorders and in how tau proteins misfold to cause several of these neurodegenerative diseases. And so I look forward to seeing you all or having you all attend then, and maybe we'll even be able to do it in person. Thank you very much.